Hey guys, so section 2.3 is called carbon compounds. And so we're going to talk through that one today. This is one of those more lengthy sections, so I'll try to keep maybe my talking to a minimum here. But kind of like section 2.1, this is going to be a more lengthy one. And we are going to spend a couple days on this because this content can be really, really tricky. And it's also going to be, for most of us, like brand new. Okay. So 2.3 is always one of those areas where I've had students struggle. And so plan to focus more of your time and energy in going over the stuff from chapter 2 on this section because you're going to need more time and energy on this section. All right, so go ahead and put down the title for me, section 2.3, carbon compounds, and we're obviously looking at a picture of a fossil here, which is something that we can date radioactively using carbon-14, which is one of those isotopes we talked about back in section 2.1. Oh, let me go back. All right, so learning objectives with this, there are only two. Identify the elements that carbon bonds with to make up life's molecules, and then explain the functions of each of the four groups of macromolecules. And so within this, we have a lot of new terminology, and that's, I think, where we run into a lot of issues with this section. All right, so we'll talk through that here together. So when we look at carbon, remember we said back in section 2.1, carbon is really important, and it's so important, in fact, that there's a whole field of science dedicated to just studying carbon, as odd as that may sound. So carbon is kind of the defining focus of what we call organic chemistry. When we talk about carbon compounds in this section, this is the same as calling these things organic molecules or organic compounds or biomolecules or even macromolecules. So the idea is that we're giving with all those, if you remember what the term macro means, these are very large molecules that we find in all living things and that play really important roles in our cells. So you think about our cells as being something that is, it makes us up and there's a lot of different jobs being done there. Well, the things that are actually going to do the jobs are these molecules. The cells themselves are basically structural components, right? But they're holding within them enzymes and proteins and other carbon-based molecules here that are actually doing the chemical reactions, okay? So when we look at carbon, carbon is really important for all living things. We know that it's a major component of all of these molecules we're going to look at today. And what you're seeing here on this slide is really examples of carbon-based molecules. Again, you can see we have covalent bonds in all these places. All these single straight lines or double or triple represent those shared pairs of electrons. And so these covalently bonded molecules are super strong. They're very resilient in the body. They don't dissolve in water. Some may have slight charges, just again, like water based on unequal sharing of electrons. So they could be polar. But for the most part, they're going to kind of stay together. Most of these molecules that you're seeing here are not actually impacting living things. The only one that's kind of an exception there is this one down here on the left, which is methane. We talked about that in terms of the greenhouse effect and climate change in chapter 7, back before fall break. Okay, feels like ages ago now. So when we look at these molecules here, all right, there are some defining features that we can look at. So again, the number of bonds. So this one here is a single bond. It's just one pair of electrons being shared. This one here is a triple bond because there are 3 lines. That would be 6 electrons or 3 pairs of electrons, 1 per line being shared. And then this here is a double bond. So you have 2 pairs of electrons being shared per line or 4 total, okay? There are different structures that we can look at here in terms of shapes that these carbon atoms can form based on how they interact with other atoms. So we have chain structures like acetylene here, butadiene here. That's also a chain structure. Isooctane here is also very much a straight kind of chain structure. But then we also have rings. So we have these benzene rings, and they're going to be rings in the nucleotides that make up our DNA. There's going to be rings sometimes in the proteins that make us up. So these ring structures also provide a lot of functionality. And then again, we have these branching chains. So like this one, isooctane that I mentioned before is definitely an example of a chain. This is a branching chain because if we just looked at the carbon atoms themselves and we kind of drew a line through those, right, you would see we have this chain type structure there, okay? So what's important to take away from this slide, carbon can form lots of different structures with lots of different atoms of different types of elements. There we only looked at how carbon interacts with hydrogen, but throughout the rest of this section, you're going to see lots of other examples of different elements that carbon can interact with, okay? So macromolecule is a term that I used a few minutes ago in talking about what these things are that we're looking at here in this section. Again, macro means big or large, so we're talking about large molecules. We're talking about molecules that are much bigger than a single water molecule or much bigger than a sodium chloride or salt ionic compound, okay? These are going to be much larger. These are typically primarily going to be composed of covalent and very strong bonds, okay? So one of the key takeaways for this, we're looking at large molecules that form through a process called polymerization, which is kind of a mouthful, but we use that term to represent the formation of these really big molecules because we're forming what we call a polymer. So if you remember our Latin roots, poly means many, and mono is the other term that we're seeing up here now on the screen, which means one or single. So when we talk about polymers, the term polymer means we have many monomers that are linked together, okay? Many monomers that link together. Monomers, on the other hand, are just identified as the single units, or sometimes we call them the subunits, that together interact in different forms and with different types of bonds to form these large polymers. So oftentimes we like to talk about this as like Legos. My two kids love Legos, being that they're 6 and they're 3 years old. Like Legos are a constant thing in our house. And so when I think of forming these polymers, and you can kind of see on this image these shapes that we're looking at here too, right? I mean, these are all similar in shape, but they're linking together in one specific way to form this larger polymer. So here down at the bottom is my polymer, and here up at the top are my individual monomers. All right. So for each of the 4 groups of carbon-based molecules we're going to look at, we're going to identify what's the name of the monomer, what does it look like, and then how do those monomers interact to create these larger polymers that generally are the ones that are going to play a functional role in our cells. Okay. So make sure you understand mono, single units, poly, many. So polymers, many monomers. Okay. Make sure you've got that jotted down. So the 4 groups that we're going to talk about, the ones that I said we're going to identify for these monomers and polymers, they're listed in the order here in which we're going to look through them. So if you would get the first bullet here, this part, like the heading in that first bullet, but the ones that are going to follow here, we're going to go through these in that order. So I don't know that it's necessary for you to write down the individual list, carbs, lipids, proteins, and so on this slide. Okay. But there are 4 basic groups. There's no more. We're not like leaving anything out. We are doing the 4 that we have. And you go on and you go on to college and you decide you want to get into the medical field, or you want to become a nurse, or you want to do forensic science. This is one of those things you get to come back and learn more about. And they're going to teach you the exact same stuff in a little bit more detail, but they're still going to talk about the same 4 groups. All right. So these are carbohydrates, lipids, proteins, and nucleic acids. All right. And these all have important roles in living things. There's not one that's more important than other. And for all living things, we need all of these working in combination, working well for survival. Okay. So when we start with carbs, let's look at those first. I'm guessing if there's one group that you've heard of carbs, and not necessarily that you've heard about it in science, but you might have heard about it from a family member at home, because sometimes I know we have individuals that decide to go on these low carb diets, or they talk about carbs as being bad for you. In reality, carbs are not bad for you. We all need carbohydrates to function. It's, again, this is, and one of the things you're going to hear me say in this section is that there's a balance for all of these things. We all need carbohydrates. We all need lipids, which are actually fats. If you lack any of those things entirely, you're going to run into issues with your cells functioning properly. So all of these are still necessary. It's just the quantity that you have may need to be kind of varied. Okay. So carbohydrates, here's your definition. These are polar molecules. They're made up of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. So what I mean by that is, if you look at a molecule that is a carbohydrate, like here on the screen, we have a molecule of glucose, which is like the primary example of a carbohydrate, maybe the best one we could look at. If we were to count up the numbers of carbons here, hydrogens and oxygens, For every one carbon, there would be about one oxygen, and there would be about double the amount when we look at hydrogen. So carbons, I have 1, 2, 3, 4, 5, 6 carbons in this molecule. I have one oxygen, 2, 3, 4, 5, 6 oxygens. And then if I go on and count my hydrogens, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, which is exactly 1 to 2 to 1. I've got 6 carbons, 12 hydrogens, and 6 oxygens. So we tend to see this ratio happening in carbohydrates, and we do not see this in other types of molecules. So that's unique to carbohydrates. When we think about the word carbohydrate, right? To me, I think carbo meaning carbon. And if you think about what it means to be hydrated or dehydrated, we're talking about water, right? And water is H2O. So for me, the way I can remember which elements make these things up is to think about the actual word part. So hydrate meaning water, hydrogens and oxygens, right? And carbo meaning carbon. So those are the only 3 elements we see in these super complex molecules. Living things use carbs primarily for 2 purposes, either for short-term energy or for structural support. We can also store energy for like a small window of time in the form of large carbohydrates, but for the most part, carbs can't store energy longer than about a day, 24, maybe 36 hours. That's about as far as you're going to get, okay? So within our carbohydrates, we have monomers and we have polymers, and what you see in this image here is an example of both of those. So we have starch down here, and if you look at the size of starch, right, starch is a fairly big molecule, and this, by the way, is only part of starch. We're looking at a very small segment. In reality, these chains go on and on and on and on and on and on for a very long time or a large distance. So we're oversimplifying it here to make it a little bit simpler for you to understand. If you want to draw this or kind of represent it, not necessarily in drawing those hexagon shapes because those get kind of tricky, but you could just do some circles kind of attached to each other so that you're representing exactly what this thing looks like. Glucose, as I said, is a good example of a carbohydrate. It's the one we tend to talk about the most because glucose is the one that we use in our bodies to generate energy. It's what we obtain when we consume food. It's what plants make through photosynthesis. It is that thing C6H12O6 that you might have heard about before. So glucose is the monomer in this example. All right, even though glucose has all of these parts here, glucose is actually the monomer because if we look at starch, I have 1, 2, 3, 4, 5 glucoses linked together. And in this scenario, they're all linked with covalent bonds, and I can tell that because I've got these nice straight connected lines. Okay, so we're sharing pairs of electrons there. But I have 5 glucose monomers that link together to form that starch polymer. Because these are carbohydrates, we give these specific terms for monomers and polymers so that we can refer to them more easily. So glucose and other types of monomers of carbohydrates are called monosaccharides. That's how you say that. Starch and other types of polymers, which we'll look at on the next slide, are called polysaccharides. And I believe on our Latin roots we talked about, or you had to memorize at one point, this Latin root, saccare. If you did not, I can't remember for this year if it was on there or not, but if you did not actually review that, saccare means sugar. So when we define these, okay, polysaccharide means many sugars and monosaccharide means single sugar, which is exactly what we're seeing. A single sugar is like this. A single sugar has a ring shape. When we link them all together in these big long chains, then we have a polysaccharide, many sugars together, okay? So let's look at some of our examples of polysaccharides. So polysaccharides are, again, they're polymers of monosaccharides. They include starches and cellulose and glycogen. And notice what they all kind of look like. Again, this is very much oversimplified. These would continue on. All right, these chains would continue to move on towards the right. But what's interesting is that these all kind of form these nice, real organized chain structures. So polysaccharides differ based on their chains of monomers. Most of them, if you notice here, take on a very similar shape to the glucose that we just saw on the last screen, and that's because they are glucose. We have Glucose here and here and here and here. But what's changing in these examples is how they're actually linked together. So let me erase those circles that I just made so that we can see those differences. So the differences I'm referring to are right here. So I have these connective pieces here, right? I've got oxygens that are all pointing down for these two, for both starch and glycogen. These are all structured the same, but the one that differs here is cellulose. Notice here, I have some of these that actually point upwards, these two specifically. So the way that we're kind of orienting and changing these glucose molecules determines what their functions will be as part of these molecules, these larger polymers. So glycogen is a polysaccharide found in animals. We store it in our muscles and in our liver, and that glycogen plays a role in energy storage. So what that means is we can take glycogen and we can create glycogen molecules from the food that we consume, from the glucose that we eat. We can store it in our muscles and liver, which is a perfect spot because if we need to use energy, chances are we're going to need it for one of those two types of organs or organ systems. So what's unique about glycogen, again, is really those two parts, animals, in that we're using it for energy storage. Starch, on the other hand, although it has the same general shape as what we see for glycogen, it is structured slightly differently. Some of these actually twist in these chains, and some of them are really just linear. But starch is made only in plants. We don't have the ability to make starch, but in plants it serves really the same purpose. It can store energy, again, only for a short period of time, though, like we're talking about a day or two. Beyond that, if we still need to use these things for energy storage, your cells will actually take them, break them down, and turn them into a different type of molecule called a lipid, which is a fat. Cellulose up at the top. Cellulose is also found only in plants, but in plants it serves a different role. So here it makes up the cell walls of plants, which is something that animals just simply don't have. Cell walls in plants provide some rigidity. They allow them to stand upright. They provide a little bit of protective kind of function for them. And in that role, because animals don't need all those things, right, those cell walls have to be kind of strengthened by cellulose. We know cellulose as something called fiber. And if you've ever heard of products like Fiber One or Metamucil, or I don't know, there's like a ton of them out there, I think. But older people tend to have issues sometimes with digestion. Fiber, or cellulose, as we know it in the biochemistry world, is not something that we have the ability to break down, and it's because of these unique bonds right here. We don't have the chemicals, the enzymes that we need to be able to actually break those bonds. And so essentially, if you consume and eat cellulose, it's going to come out in roughly the same way that it went in. All right, so like think about if you've ever had corn, the outside of those corn kernels is made up of cellulose. Bread that's like very much just ground wheat, it's not processed much, like whole wheat products. Tend to go in and they come out in the same way. Not saying that you don't digest it, but the cellulose molecules that make it up are going to come out the same way. So in a lot of ways, fiber is important for maintaining digestion, and it helps to kind of stimulate the number 2 function for humans, which is disgusting maybe to talk about, but is certainly important for human health, right? For some of us more than others, I guess. Chitin at the bottom is another type of carbohydrate. It's different from the others. You can see the molecules that make it up are slightly different, and the bonds that they form here are also slightly different. There's a nitrogen in this one, which we don't see in the other ones. But chitin is important in animals, specifically in things like beetles. Like I always think of crunchy insects, things that if you step on them, they'll go crabs and lobsters, all those exoskeleton structures, the shells and the protective pieces on the outside are all made up of chitin, okay? So that's also a polysaccharide. All right, so switching gears here a little bit. And I should mention also, by the way, if I skip back one slide, glucose is not the only monomer of a carbohydrate. There are other types. There's something called ribose that we'll talk about in the form of RNA. That's also a ring-shaped monosaccharide. There's deoxyribose, which we find in DNA, which is also ring-shaped. There's galactose. There's a lot of other variations of this, and they all tend to end with ose. There's also something called sucrose, which is actually a disaccharide, which means there's 2 linked together, and that's a fructose and a glucose that are linked together. And if you've ever heard of fructose, that's what we see in high fructose corn syrup. That's also a monosaccharide. Glucose and fructose, when you link them together, they form sucrose, this little disaccharide molecule, which is the thing that you eat when you eat sugar. So if you've ever baked anything with sugar in it or added sugar to your sweet tea, right? That sugar that you buy, the thing that's labeled as sugar at the grocery store or in your cabinets at home, is sucrose. It's glucose and fructose linked together. Okay, it's just a disaccharide. So really simple molecules, but again, things that we see really on a daily basis. All right, so switching gears is our second group. This one I think is more complicated than carbs. So these are lipids. These are more complicated because they're a little bit more diverse. So lipids are non-polar molecules. Carbohydrates were polar, which means that sugar likes to dissolve in water. Lipids, on the other hand, won't dissolve in water because they have no slightly charged areas. These are made up of carbons and hydrogens and oxygens, just like what we saw in carbohydrates, except they have much less oxygen than carbs. So here, this item here would be important to jot down so that you kind of remember this as a major kind of defining feature of lipids. So much less oxygen as compared to carbohydrates. In this case, the ratio of hydrogens to oxygen is much greater than 2 to 1. So if you look at the image here on the right, the molecule that you see here, this is one giant lipid molecule. It's huge. And when we look at the size of this thing, one of the things that you should be able to see right away is I've got a whole lot of carbons here lined up down the middle, and I've got a whole lot of hydrogens on either side, but then I have only like 6 oxygens up here, right? I have an oxygen here, I have an oxygen here, I have 3, 4, 5, and 6, and that's it. So I have a ton of hydrogens and a ton of carbon in this one. So we use lipids in our bodies to store long-term energy in the form of fats. We also use them in our cell membranes. They're really important there. And we also use them as precursors for steroids, meaning we use them to create steroids and other hormone molecules. All right. So when we look at these types of molecules, this one here that you're seeing on this screen is called a if I can write this down, triglyceride. Okay, a triglyceride molecule. So this is this whole thing right here. Triglyceride is made up of, like you see up at the top, this orange part, which is called glycerol, and then 3 fatty acids. There's 1 fatty acid here, 2 fatty acid here, and 3 right there. So fatty acids are actually going to be the monomers when we look at lipids, and we'll talk about those on the next slide also. But notice with these fatty acids, they can have some different shapes and structures here. So I have some areas of these fatty acids where I have a couple double bonds, and then I have some other areas where I just have single bonds. That makes up the majority of it. These things where we have double bonds are what we call unsaturated. And so an unsaturated fatty acid, if I can get to the part where it's labeled here, unsaturated fatty acids are going to be important for us, especially on a dietary level, because these are going to be things that are healthier for us. These are the healthier types of fats or lipids that we're going to consume. What makes them unsaturated is the presence of that double bond. And what that means is that when these things are kept at room temperature, they don't solidify. So we're talking about like the olive oil that you see here on this picture. We're not talking about animal fat or butter, which we know if we leave at room temperature, just set it out on the counter, it's going to remain in that solid form. Or if you fry bacon on the stove and then you let that bacon grease sit in the pan, it becomes solid, right? It's not like rock hard solid, but it's not a liquid, right? You can't just pour it off if you let it sit there for a while. Because animal fats are not unsaturated. Animal fats are solid at room temperature, which is why our fat in our bodies is solid at room temperature, right? We don't store fat as oils. We store it as a solid substance, okay? So unsaturated tend to be liquid at room temperature because they have double bonds. Saturated fatty acids and saturated fats are more solid at room temperature, and that's because they have only single bonds. So they pack together more tightly. Those atoms and those molecules kind of align, and they can form these solid structures, okay? So if we go on here and look at more specifically these fatty acids, right? On this slide, by the way, I would make sure you get every bit of this because all of this is important. And these are not vocab terms, but I put them in italics because I know you should know those, okay? So I mentioned those fatty acids that we just talked about, which can be unsaturated or saturated, are the monomers. Okay, so the monomer of a lipid is a fatty acid. All right, and those are those big long chains of carbons and hydrogens. Lipid polymers that contain 3 fatty acids bonded to a glycerol are called triglycerides, and this is again how we store fat in our bodies. So this is body fat in humans or in animals, which sounds like it's fun to write down, right? Body fat. So that's the way that we store really energy long term. All right, and that's the purpose there. So the idea is that if you consume a lot of food and you're not burning it off, right, you're creating a lot of body fat, which is essentially just stored energy. So if you start using or doing things that requires the use of energy, you're going to start to burn off that body fat. Okay. If, however, your use of your energy exceeds the amount of food you're taking in, right, you're not storing much as body fat. You're not storing energy for the long term. You're basically using it as you consume it. So in addition to storing energy in the form of triglycerides, we also know that cell membranes are made up of another really important type of lipid called a phospholipid. And we're going to talk about these a whole lot more when we get into cells in chapter 8 in a few weeks. But phospholipids in cell membranes look like this, which you see down here. They're super complicated structures. The phospholipids are like the little pink spheres here that you see. Let me move to this. Little pink spheres, and then the little lines coming down. The little jaggedy lines, those little greenish lines, are representing the 2 fatty acid tails attached to that what we call a phosphate head group. So phospholipids have 3 parts. They're made up of a glycerol molecule, just like our triglyceride, 2 fatty acids, not 3, and a phosphate group, which the formula for that is PO4 with a little negative charge. And phosphate, by the way, is going to be something you're going to hear me talk a lot about this year. This is a molecule that we find in many, many different parts of cells and has many roles in a cell. Okay, so the phosphate group kind of determines the orientation of these phospholipids. And like I said, we'll talk about that when we get to chapter 8 in a little while. Okay. Cholesterol is another type of lipid. This is used to create hormones and steroids. It does not contain fatty acid monomers. And actually, cholesterol, if I scoot down here, is this molecule right here. So this over here is actually our cholesterol molecule. Okay, so notice there are no fatty acid chains with that. What we're looking at is several rings and some things kind of coming off of that, a branching chain there. But this is classified as a lipid because it is nonpolar. It has a ton of hydrogens and a ton of carbons, and really it's got only one teeny tiny little oxygen atom right there. All right, so it meets the characteristics and the qualifications for a lipid. Its job, though, is very different. It doesn't provide energy. It doesn't provide structure in the form of a cell membrane. What it's used for is to create hormones and steroids and other things that kind of, as a teenager, for y'all, kind of make you a little bit crazy, and me too sometimes, right? All right, switching gears again, our third group is the protein group. That means we're at least halfway done, so that's good. So proteins, vocab term here. These are polymers made up of monomers called amino acids. So this is important here. When we talked about carbohydrates, our monomer was a monosaccharide. Our polymer there was a polysaccharide. When we talked about lipids just a second ago, our monomer was a fatty acid. Our polymer was some different examples. There were triglycerides and cholesterol and phospholipids. When we talk about proteins, we don't really have a specific name for the polymer of a protein. We just call them proteins. Sometimes we call them polypeptides. So I'll jot that down. Polypeptide. So a polypeptide is an example of a polymer of a protein. It's just another name for a protein polymer. But a polypeptide and a protein, or any type of polymer that we could refer to in this category, are made up of amino acids. That's the monomer. Here we're introducing now interaction between other types of atoms from other elements. So now we're going to incorporate nitrogen, which is present in every single monomer, every single amino acid that makes up a protein polymer, and sometimes also sulfur. So that's what S stands for if you weren't quite sure. The general structure of an amino acid is right here. You do need to jot that down. Okay, the way that I normally write it is something like this. So I have a central carbon which is there in the middle. And coming off of that, I have 4 bonds because remember, carbon has the ability to bond with 4 different things. It has 4 valence electrons, like we talked about in section 2.1. On the easiest parts, we have a hydrogen up here, and that's consistent for all of these groups. We have a group here on the right, which is called a carboxyl group. That's COO with a negative charge or with an H, but it's a carbon and a couple of oxygens. And then we have our amino group here, which is how we got the name amino acid, and that's NH3 with a little positive charge. When these things link up to form a protein, they link up to the right and to the left. All right, so these go in both directions that way, and they form like big chains like that, and then they fold to create the actual protein structure. The last component here is R. Now, R, if you know anything about the periodic table, R is unique because R is not a symbol of an element in the periodic table. R is something that we call a side group. Now, you can think of it like, okay, it sticks out the side, right? It sticks out in the bottom part of this amino acid. But the side group is unique because it's what allows these amino acids to be different from one another. So in reality, we have a total of 20 different amino acids, and the only thing that makes them different is what we see here at this R or this side group. Every other component here, this whole part of this amino acid is going to be exactly the same regardless of which amino acid we're looking at, regardless of which of the 20 we're looking at. It's the R group that defines them and gives them some physical and chemical characteristics. Okay. So let's see. I've got amino and carboxyl there. Let me erase the other stuff I've put on here. So here are some of those labels, all right? So it would be good if you label each of these amino and carboxyl, right? And then, of course, that side group there. What they're showing you down here is how we actually form something called a peptide bond. And a peptide bond is the very last type of bond that we have to look at. And I've got those coming up here in a second. But again, they're going to link together left to right. And you can see here, if I start with 2 separate amino acids, I'm linking my carboxyl group to my amino group. And as a result, I get a waste product, 1 molecule of water that gets released. And these 2 then are covalently bonded. Okay, so that's kind of how that bond forms. And that's, again, going to be our 4th and final bond for this chapter. And I'm getting there the slide after next. Okay. So hold off for that for just a second. All right, I'm going to move myself over here. All right, so proteins are the most varied of all the biomolecule groups. They can be kind of tricky for that reason, but in other reasons, in other regards, I think they're simpler because they just have this one amino acid structure and they're just called proteins. Like I mentioned, all living things, we have 20 total amino acids, and that's for everything from the smallest bacterial cell and even viruses, which remember we're considering non-living, protists and fungi and plants and animals and even humans, right? We all consist of just 20 amino acids. In humans, we can create 11 amino acids, 11 of the 20. Our cells have the ability to do that. But then that means that we're lacking 9. The remaining 9 are considered essential. What that means is that we have to consume them in the food that we eat. So it's important, again, like I said earlier, there's a balance for all these things, right? Our diets need to contain lots of different types of foods on kind of a daily basis to make sure we get all the essential amino acids and vitamins that we need for survival. And so it's important even for individuals that may be vegetarian or vegan. We know proteins in a lot of animal products. So for them, they have to obtain protein in other sources. Soybeans have lots of protein. Beans in general have lots of protein. Peanuts and other types of legumes, almonds and those types of things. Fish, if they're not full vegan or vegetarian, has lots of protein also. So it's really important that we all obtain kind of a basic amount of all these things in order to allow our cells to do what they need to do. The picture that you see here on the right is an example of all of the 20 amino acids. You can go through and count them if you really wanted to. I will not. But they're organized based on the different chemical properties that they have. So the ones in blue here, if you can see it, these are amino acids with electrically charged side chains. So these are R groups that have charges. So there's a charge here, 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 and here. Here are amino acids that are polar, but they have uncharged side chains. So these are polar side chains, polar R groups, but they're uncharged. So they're not full ions. They may have partial charges, kind of like we saw with water as a polar molecule. These are special cases that don't really fit into other categories. We have a ring here. We have a super simple amino acid in glycine with just a little hydrogen atom right there. We have sulfur in this example. And then down here at the bottom, we have hydrophobic side chains, and that's a term that we'll come back to a little bit later, but that essentially means that these guys are kind of afraid of water. Okay, so the way that they interact in a space where we might find water is going to be kind of unique. So again, these are your 20 amino acids. You don't have to be able to do anything with them at this point. You just need to know that there are 20. Okay. By the way, all their names are up here at the top if you were interested in those. But again, you don't need to know those right now. All right, so I mentioned that proteins form our last type of bond, which is called a peptide bond. And that's what we're looking at here. So a peptide bond is really the same as any other type of covalent bond. What's going to make it unique is that we only see them in proteins. That's it. So we're still sharing pairs of electrons. It can be unequal sharing like we saw in water, or it could be fully equal in how they share. But those covalent bonds, those peptide bonds, as we're calling them here, are important enough that scientists gave them their own special name. So this is where we get the term polypeptide for a protein polymer. So if you think about the meaning of that, right? Poly meaning many and peptide meaning the bond that we're forming here. So we've got many peptide bonds. A protein, by definition, consists of many, can consist of many polypeptides. It could just be a single polypeptide chain where we have lots of peptide bonds linking these amino acids. Or it could be something like this molecule we see here, which is actually called hemoglobin. And we have 4 distinct chains of polypeptides. We have 1 polypeptide here, 2, 3, and 4. And those, all 4 of those come together and interact to form this thing called hemoglobin, which is really important in your red blood cells because it's how we transport oxygen through our blood. Okay, so obviously a complicated protein there with a very specific job. The way that we organize our amino acids determines how a protein molecule functions and how it's also going to be structured. Okay, so it's essential that we get kind of that order correct. We can use proteins for a variety of different things. If you were in my class and we were looking at this slide, I would say for this part, jot down a few examples. You don't have to do all of them, but I like to give you kind of a broad spectrum in terms of what these things are doing. So they can be used as enzymes for chemical reactions, and that'll be the focus of section 2.4. We'll talk about that next time. They can be used to transport things into and out of a cell. They can be used as chemical messengers. Again, they can also be hormones similar to the lipid molecules we saw a couple slides ago. They can help with muscle movement. Obviously, protein, usually when I say it, people think muscle. So they certainly help with muscle. Immune support and protection, antibodies are coming from proteins, right? Hormones, gene expression, nutrient storage, and so on and so forth. So there's a lot of variety here in this group. But the main kind of takeaway, I would say, with proteins is to understand that these are what are really doing the work in our cells. So I often say these are the workhorses of our cells. Anything that needs to be done, I can guarantee is going to be done by a protein. Okay, so these are super important in that way. The last group we have to talk about is nucleic acids, and I only have 1 slide for these because we spend a lot of time on these later in the year. All right, when we talk about DNA and RNA and genes and transcription, translation, we don't need to do any of that right now. All that we're going to do is talk about kind of the basic chemical properties of these. What are the monomers? What are the polymers? And what is their function? Okay. So nucleic acids, this is the name of the polymer, are made of nucleotide monomers. So the pieces that link together are nucleotides. This right here is a whole nucleotide. This is a large monomer structure, right? There's 3 components to it. We have a nitrogen base, which is the green part. We have a sugar, which is deoxyribose or ribose. And then we have a phosphate group. Remember, this is the thing we also saw in phospholipids in that lipid section a few slides back. There are 5 different types of monomers. There are adenine, guanine, cytosine, thymine, and uracil, or also known as A, G, C, T, and U. And we'll talk about all of those later on in the year also. But the only real examples that we have for nucleic acid polymers are just DNA and RNA. And certainly, I would expect that you know a little bit about DNA, if not also RNA a little bit. But the function of DNA, if you were unsure, we're using these to store information, and then RNA helps to transmit genetic information. It provides a temporary copy or a code of a gene in DNA that allows us to make a protein that then carries out a certain function, right? So all these molecule groups are kind of interlinked, and they all play, again, important roles in our cells in that way. Okay, so make sure you've got, for this one, the monomer name, the polymer names, and then certainly what their function would be. Okay. As always, I think I've got another video to attach with this, and I'll post that on your agenda. But other than that, if you've got any questions, let me know, and I'll see you next time for the last section, which is enzymes. All right. Take care, and I'll see you later. Bye.